Good morning. Now we're going forward the fifth chapter of the book in the first video. The problem to fix is how to compare biological sequences. And we are going to describe a set of algorithms that use the dynamic programming paradigm to do so. The first question that we wish to investigate is how do we get a meaning out of a sequence or out of a sequence comparison? The central dogma of molecular biology claims that we go from DNA to RNA. The RNA is transcribed into amino acid, and chain amino acid constitute a protein. But we have non-ribosomal peptide synthesized that allow us to obtain peptides out of a DNA. One example is the NRP synthesized. This protein is composed by 10 domains, and we wish to compare them. On the first side, they look like very dissimilar. I have only had 3 conserved columns. What do I mean by conserved column? I'm going to compare amino acids by position. The first position is the first line is Y. The first position is second line is A. And the first position is the last line is I. In this column, I have L, L, and L. In this column, I have K, K, and K. In this column, I have G, G, and G. So out of this consistent number of letters, I have only 3 columns containing the same character. But I can use a trick. I can put a minus. This is called a gap. Putting a single minus, inserting a single minus at the beginning, I'm going to have not 3, but 11 identical columns. This one, this one, this one, this one, this one, and going forward. So a trick is to put the gap in the appropriate positions. If I'm going to put or to insert further gaps, I'm going to obtain 19 conserved columns. And I paint them in red. Now I'm going to show you the non-ribosomal code, but this is not a bioinformatic issue. So we are not going to stop for a long time on this one. As an example of the importance of the application of bioinformatics, we are going to report the cystic fibrosis. that is a severe inherited disease. We have a gene, a mutation in a gene, then when happen in both chromosomes, give a 25% of likelihood of the individual of being affected. When happen only in a single chromosome, the individual is a carrier, but he is not affected. The point is that when they now research for the gene, they identify the CF gene, cystic fibrosis, in a small region on chromosome 7. The point is that one of these genes was similar, how do they know it? To ATP binding proteins that act as transport channels responsible for secretion. Now we had some hint, and we know why do we want to compare genomic sequences. We are not going to go deeply on this part because the subject of this course is bioinformatics, is not biochemistry. This video is concluded and we are going to meet in the next video.